Okay, well, good afternoon, everybody. Thank you very much for joining the NC3R's 3Rs Prize Award for this year. I'm Kevin Shakespeare. I'm the chair of the NC3Rs and also Pro Vice Chancellor at the Open University in the UK. And it's always a great pleasure to introduce this session where we celebrate some great science that's been done that's relevant to the mission of the NC3Rs and all of our people on the call who are doing work in the 3Rs research field. The prize is made possible by the long-standing sponsorship of GSK, and we're going to hear in a few minutes from Mick Mahon, who's from GSK. But just to thank them, they've supported this prize since 2005, I think continuously. So it's a great commitment and has recognised decades now of really excellent work in the field that's gone on to have a great impact on the 3Rs. So for those of you that attended last year, I think we were in the early stages of the first lockdown in the UK from COVID, and it all felt very new, meeting together over Zoom. So we're continuing this year, and probably finding it's allowing more people to join the meeting from around the world and hear about the work. So there are benefits as well as the disadvantages of maybe not bumping into people and having those sort of more informal discussions. But I hope you enjoy and get what you want from today's sessions. We're going to have a presentation from our prize winner in a little while, and we're going to have a session at the end for question and answers. Just to say the way we're going to do the questions and answers, if you can pop them in by text into the Q&A button, which should be down towards the bottom on your Zoom screen. And then staff from the NC3Rs will select and read out questions. So you won't actually have to appear yourself to ask the question. I should also say that we are recording this session. And so you can also let people know if they want to watch it at a later date. Yep, so the prize, as I said, been running for many years. We're looking to recognise an excellent piece of primary research that's been published in a peer-reviewed journal over the last 3 years. It's open to any researcher in academia or in industry. And we give the award to a principal investigator or a team or any other of the nominated authors. So in its 16th year, we brought together a prize panel with experts from across the world, and I'd like to take this opportunity to thank the panel members for a really diligent job. We had it's always really difficult to do these decisions, but we had an excellent long list and short list of new papers. We went through that, and all of them were excellent. We looked at 4 main criteria for deciding on the prize: the actual or the potential 3Rs impact that the research could have, the quality of the published research, particularly transparency and robustness of the studies. We asked the applicants to tell us about the 3Rs implications, and so we assessed that outside of the main paper. And then we asked them what they were going to do with the prize, and also judged on the basis of how good we thought their plan was for the use of the prize money. Let me also just thank the staff at the NC3Rs who have run the competition and made it all work really smoothly and brought us all here today. So what I'm going to do at this stage is to ask our sponsor to say a few words, and GSK, as I say, have been the long-running sponsor. Dr. Nick McMahon is VP of Non-Clinical Safety. So I'd like to welcome Nick, and I think we've got a brief presentation from him.
So thank you to Kevin for the kind introduction. It's a real honour to be here today representing GSK in what's the 16th year of our partnership with the National Centre for the 3Rs for this prize celebrating excellence in global science, contributing to the replacement, reduction and refinement of animal use. I would like to thank Dr. Margaret Landy and her team in the Office of Animal Welfare at GSK for their support of this prize. GSK's commitment to this global prize, recognising groundbreaking research, is a reflection of how we view the 3 Rs as integral to our purpose of discovering and developing transformational medicines and vaccines for patients. Over the last year, we have seen the power of science to push the boundaries of understanding to transform human health at a global level. It is more important than ever to accelerate opportunities where replacement is possible and to embed robust science and welfare practices that reduce and refine the use of animals. And so at GSK, we are thrilled that through this prize, we celebrate, advance, and inspire more outstanding 3R science. I must commend the high quality of the global applicants, the diligent and challenging work of the review panel, and overall the quality of the prize winners. The winning papers are examples of tremendous bodies of outstanding high quality science and teamwork with clear 3Rs benefit. I hope this grant helps to pave the way for new prize winning work in future. I'm pleased and excited to see the prize winners that will be announced here today.
Thank you very much to Nick for the comments and for GSK for their support. So we're going to move on now to announce the prize winners, and then the main winner is going to give a brief presentation. This year we've got 2 awardees to announce. So we have the main award, which I'll do second, but before we get to that, we did also feel that one paper was deserving of a high commendation. And so we're going to reveal in a second that the highly commended paper was by Dr Jennifer Ashworth. And this paper was on peptide gels of fully defined composition and mechanics for probing cell-cell and cell matrix interactions in vitro. And Jenny's at my old stomping ground of the University of Nottingham. So many congratulations to Jennifer. One trouble with Zoom is there's no real way of showing collective praise. So assume we're all clapping if you're listening in, Jennifer, and congratulations for that prize. The panel in discussing this in this paper recognized the great potential of non-animal-derived hydrogels. We know that a lot of research is done with matrices such as Matrigel, which are derived from mouse tumors. And whilst some excellent results have been generated from Matrigel, we're also aware that there are limitations with it, both in terms of batch-to-batch variation and the fact that it's difficult to tune some of the biological and physical properties of Matrigel. So Jennifer's work is part of a body of work that's going on throughout the world that's looking at ways in which synthetic and fully defined hydrogels can replicate the benefits of Matrigel without the need for it to be derived from this mouse tumor model, and hopefully allowing us to do better science by having better control of the conditions that were presented within the hydrogel. So overall, the panel was impressed by this technology, saw a very wide range of potential applications for which it could be used, and noted that the research team's already proactive in disseminating the technology and demonstrating the potential for 3R's impact. So many congratulations again to Jennifer, and of course it was a team effort, so congratulations to all of the team that were all involved in that. So let's move on to the main winner, and I'm very pleased to announce that the winner who will be presenting today is Laura Pellegrini, and Laura's paper was human CNS barrier-forming organoids with cerebrospinal fluid production. And Laura is based at the MRC Laboratory of Molecular Biology. I won't say too much on the paper because we're going to hear about it in a second, but just to say that as a panel, we consider the work to be groundbreaking research for the neuroscience field. Exciting opportunities for understanding the problems around drug delivery and the challenges around delivery to the brain. We felt that the organoid system could be transformative in nature and had already attracted lots of interest from potential end users. So although it's early days, we can see this technology having a major impact on the 3Rs in the future. So huge congratulations to Laura and all of the team. And at that stage, I'm going to pass over to you for your presentation.
Okay, thank you so much and good afternoon, everyone. First, I'd like to thank the NC3Rs committee. It's really an honor to receive this prize and to have the opportunity today to tell you about my research. So the publication that was awarded, the title of the publication is Human CNS Barrier Forming Organoids with the Cerebrospinal Fluid Production. So what does this mean is that basically we've created, we generated an organoid model that recapitulates a barrier in the central nervous system called the choroplexus. So what exactly is the choroplexus? So it is a secretory brain barrier, and we find it at different locations in our brain ventricular system that you can see here in blue. So we find it at different location, and it is basically a structure, an epithelial structure that surrounds a connective tissue, a network of connective tissue, immune cells, and vasculature. So the cells of the choroplexus are polarized and they are tightly packed with tight junction forming this barrier. When we looked at the difference between our human choroplexus compared to mouse choroplexus or other rodents, we can see a striking difference in terms of complexity of the tissue. So in mouse, we can see that the structure of the choroplexus is much thinner compared to the human that is much more expanded. And you can see it perhaps even better here in section of brain sections at developmental matching stages of development in rat and human brain. So you can see that the choroplexus occupies almost all the ventricular space. And this perhaps suggests that it could have had an impactful role in the expansion of our cortex as well. But so why do we care about the choroplexus? Why should we study it? So the choroplexus is an interesting interface, an important interface between two fluids, the blood and the cerebrospinal fluid. And in fact, its main role is to secrete the cerebrospinal fluid. So, and it's quite dramatically doing this because every hour our brain secretes about 25 ml of cerebrospinal fluid, which is about the volume of a shot of espresso. So the cerebrospinal fluid is not only important in order to provide a supportive environment for our brain cells, but it also contains several important factors, vitamins and hormones. And the neuronal cells and the progenitor cells are exposed in contact with the cerebrospinal fluid can in fact be modulated by these factors. And there's evidence that certain factors in the CSF, in the cerebrospinal fluid, can modulate proliferation or migration of neuronal cells. But the choroplexus, as I mentioned, is also a barrier. And this basically prevents certain drugs from entering and crossing the brain. And this is a huge problem in the design of certain drugs that are, in fact, difficult to get across in the brain. But it also prevents from entry of pathogen. And there's also several immune cells in the connective tissue that surrounds and support the choroplexus. And these immune cells are also important to prevent and to basically prevent and as a defense mechanism to prevent pathogen to enter. So the other barrier that is mostly studied is the blood-brain barrier. But this barrier is a very complex barrier compared to the choroplexus, so the blood CSF barrier. And in fact, we think that the choroplexus barrier could be perhaps an alternative way to get drugs across the brain, and in fact, simpler perhaps to model compared to the blood-brain barrier. And also the blood CSF barrier is developing earlier on compared to the blood-brain barrier. So what are the current limitations and why there is a need for a model of the choroplexus? So the choroplexus is located deeply in the brain, and so this makes it challenging to dissect and anatomically challenging to dissect. It is challenging also to isolate pure CSF without blood contaminations. And often to extract CSF, we need to perform what's called a lumbar puncture, which is quite invasive. And there's obviously ethical and technical issues related also to the collection of human biopsies, especially early on in development. Therefore, also most studies that regards choroplexus biology are performed in animal models such as mouse, rat, and also macaque. So the aim of our project was then to generate a human choroplexus organoid. And in the lab, I'm in Madeline Lancaster lab, and Madeline has established a protocol to generate cerebral organoids. And in the generation of to generate cerebral organ, we start from pluripotent stem cells. We aggregate them at the bottom of the dish to form what's called an embryoid body. Then we promote the formation of a primitive layer, embryonic layer called the neuroectoderm, which then will give rise to different brain identities. But the most predominant brain identities that we form, that we see generating in this organ is in fact telencephalo, for brain telencephalic regions. But here you can see a section of one of our organoids, and you can see here in green this slightly different tissue compared to these areas which are called ventricular areas. These green areas here is in fact the epithelium of the choroplexus, and we can see it spontaneously generating sometimes in our organoid. So in order to enrich in this region, what we did was to apply local signals that are locally secreted in our brain, and they promote the generation of choroplexus in vivo. And there are several studies in the literature that show that these signals are important and necessary for choroplexus formation, for example, from Yoshiki Sasai and Edwin Monuki. So we took the studies in the literature and we simply apply different concentration, a specific concentration at a specific timing of the protocol in order to generate and enrich the organoid in this tissue. So you can see here how different these organoids are, and they are completely enriched in choroplexus epithelial tissue. What's more striking though is that this organoid can form a barrier, and they completely form these cystic compartments that are filled with fluid. So we examined this fluid, and we could see that this fluid is very similar to, in fact, the cerebrospinal fluid. So histologically also, we then compared the choroplexus organoid with human fetal choroplexus and the mouse embryonic choroplexus, and we can see the close resemblance now with the human choroplexus. We can also see the expression of specific choroplexus markers such as aquaporin channels, which are important for cerebrospinal fluid formation. And we can see that they correctly express this marker in the right location. But then to really assess the permeability and the barrier function of this organoid, we then decided to test small therapeutic molecules. And we look at this example of two very similar small molecules that have opposite permeability. And these are levodopa and dopamine. So we know that dopamine cannot cross the brain barrier, but levodopa can cross through active transport. And so when we add both of these molecules in the media, and then we retrieve them both in the media, and we looked at the fluid inside of the organelle, we can in fact only detect levodopa, demonstrating that this choroplexus barrier, it's actually selectively permeable, and not only qualitatively but also quantitatively. And we've looked at different other drugs, and we can demonstrate this also for other compounds. The other thing we tested was, in fact, the content of this fluid inside of the organoid, and we looked at different cell lines and different batches of organoid and compared it to in vivo CSF from different species, and we use mass spectrometry then to analyze the content of the proteins. And we could in fact see, and you can see it here, that we, in the top 50 proteins that we detect, the top 50 most abundant protein detected in vivo CSF are also present in the organoid CSF. And among these proteins, we can detect and retrieve several biomarkers that are in fact important for the disease monitoring, the disease and in particular several neurodegenerative diseases such as Alzheimer's disease. And you can see them highlighted here in yellow. So in terms of numbers, what we actually calculated is that the volume of a CSF that you can actually collect from a mouse embryo is around between 5 and 10 microliters. And the volume of CSF from an organoid is usually between 15 and 100 microliter. We usually make a batch of organoid. And this means that from a single batch of organoid, we can actually arrive to collect up to milliliters of CSF. And just for the visual comparison, you can see this is the volume dyed in blue, and this is 10 microliters, and this is what 1 ml of fluid will look like. So this means that for 1 milliliter of CSF, you would actually need 200, or around approximately 200 mouse embryos to collect. So this is really now changing the fact of that now we have a significant amount of volume to work with, and we can perform like several biochemical techniques that previously were really, really challenging working with such small volumes. So what is the present and future of Choroplexus organoids? So I've told you about the fact that we can use this model to look at drug permeability, and this could potentially be used as a preclinical in vitro test to test novel drugs, whether they can enter or not. We can look at specific biomarker secretion. We can also potentially do disease modeling, because we can start from pluripotent stem cell, for example, from patients with genetic mutations, and looked at different secretion of different biomarkers over time. What we also looked recently, and this was a recent publication, is also the pathogen entry function of this organoid model. So we looked at, in particular, at SARS-CoV-2, and we could demonstrate that SARS-CoV-2 can in fact infect the choroplexus and cause barrier damage and leakage of CSF protein in the media. In future, we are also planning to use these organoids for comparison and species comparison. And in fact, in the lab, we are very interested in evolution as well. And it would be very important to look at also like the evolution of the choroplexus and the comparison of CSF components. But what I'm also interested in, how I would like to use this prize for, is various other functions of the choroplexus. And in particular, I'm looking at the moment at the immune modulation role of the choroplexus by seeding immune cells in the organoid and looking at the penetration of these immune cells in the tissue. I'm looking also at the circadian cycle and the implication of choroplexus in sleep, in day-night cycles. But most of all, my project, which is going to be a little bit outside of the focus of the lab, is on the role of choroplexus in clearance, and in particular amyloid uptake. So there's some evidence that the choroplexus express some markers and transporters that can uptake amyloid proteins, and this could be one of the early mechanisms that is damaged in early Alzheimer's diseases. So this would be a very interesting model to look at early stages of neurodegeneration in a context that is free of complex other biological mechanisms such as aging. So with this, I would like to thank my lab, which is a very supportive and wonderful group of people, in particular Madeline, for allowing me to work on a project that is a little bit outside of the focus of the lab. I would like to thank my collaborators, in particular Claudia Bonfio, for the NMR measurements that were indispensable for the study of the drugs in the permeability screen. and the LMB facility, in particular the mass spec facility. And I would like to thank you all for your attention.
Thank you, Laura. That was a really fascinating talk. So we've started getting some questions through. So if you're okay, I will start firing them off. So the first question we have is, could you tell us a bit more about the types of mechanistic studies or basic biological questions that the organoid models could be used to investigate?
Yeah, sure. There's various, interesting potential biological question that we can answer with this. And for example, we are looking at the oscillatory behavior during circadian rhythm, because it's been reported recently that the choroplexus is a part of the circadian clock, so it's an important regulator of this dynamic, so it could potentially oscillate and regulate turnover, protein turnover. So we're looking also at this in collaboration here at the LMB. There's also, what are we interested in is also the secretion of human-specific proteins, and we can compare what's secreted in the CSF with other CSF produced from other species. So we've already identified some human-specific proteins that could have, for example, be particularly secreted at specific stages of development, and therefore contributed to the early brain expansion in the human cortex. And then, yes, of course, the regulation of the uptake. This is a very big problem actually in the field. It's understanding the mechanism that actually drives protein uptake in the choroplexus, and there's probably more than one mechanism involved, but the machinery that is actually regulating uptake of big proteins or peptides is not completely understood in the choroplexus, especially in human.
Thank you. Sounds like there's lots of potential for extra 3Rs impacts. So we have another question from a different tact, really. So it's, how does aging affect the choroplexus structure and function, and can you model these changes in the organoid system?
Yeah, that's a very interesting question, and so it's there's some evidence that choroplexus during aging would start to become more leaky and would decrease the secretion of certain of CSF, and in particular of certain proteins. But we don't know whether that is the cause, for example, of certain neurodegenerative diseases, or if that's a consequence and that comes in later. So it's something that we're trying to look in our system. Now we're growing these organoids for very later, for until like hundreds of days, and we're seeing some early changes, and so we're investigating this at the moment with the developmental time course and single cell RNA-seq analysis of later time points.
Thank you. Our next question is around the challenges associated with working with the organoids. So can you tell us how easy they are to work with, and is there any risk of damaging the sacs of CS-like fluid? Yeah, so I guess it requires some practice, but they're not any more difficult than other organoids, I would say. It's just because the epithelium that surrounds this cyst is a single layer of cells. Obviously, they tend to be quite fragile, so they need to be moved with a bit of care, but they're quite easy to and quite reproducible to generate, because the addition of these small molecules that we add, BMP4 and this Wnt signaling activator, it's a very potent signaling mechanism, so they're sort of quite strongly recapitulated. Yeah. Thank you. And another question about the viability of the organoids. So how long can they be maintained in culture?
So we've collected fluid from organoids up to 140 days, but I would say you can probably keep them even longer.
Excellent, thank you. So It's a question about uptake, really. So if a rodent model user wanted to switch to an in vitro system like yours, are there any potential barriers that they would be faced with in establishing the system in their lab?
I mean, I wouldn't say there's a lot of now kit that we use also to make organoids, and there's a lot of simplified sort of protocols that you can use to make organoids. So I wouldn't say it's particularly challenging. I would say it's more challenging to work with animals for sure. It depends on the question, obviously, that you are interested. If you are interested in a question of the vasculature surrounding the choroplexus, that's probably not the right model because we cannot model that. But if you're more interested in the epithelial part or the stromal part, that's definitely a good model to work with.
Thank you, that's really interesting. So we have a question about whether you're able to disrupt the permeability, the tight junction barriers of the organoid to mimic inflammatory disease.
So the immune component in inflammation is not present in our system, but we can add it. We are doing this at the moment, like by seeding immune cells, and this is possible. For example, with the SARS-CoV-2 study, we were able to model the leakiness of the barrier and the damage just by adding the virus. After a few days, the barrier would already start to leak, and we saw mislocalization of tight junction protein. So that's possible to model, but it would depend also on the type of disease that you are interested in modeling and what component is causing the leakage.
Thank you. So we had a follow-up question about taking up the model. So someone's asked, you've mentioned that the organoid technology is licensed to a company. So the question is, how would one go about working with the model?
So, I mean, the paper is published, and all the protocol is published, and you completely can use it. The company will just have its own right to, I guess, produce a kit, but I'm not entirely sure what is actually is still ongoing. So we are not sure. It's still a very long process for the approval of the patent and everything. So I am not entirely sure what will be the result of that, but I am sure that everybody it's possible to work with this system, and there's no restriction for that.
Thank you, Laura. Perhaps we could encourage people to contact you directly if they've got questions about how they could.
Can also update with the situation for sure.
So a question about the ongoing plan. So it's really what inspired you to move into the Alzheimer's disease research field.
So during my PhD, I've worked I've worked on Parkinson's disease, and I've always been interested in neurodegeneration as well. So I've sort of decided to merge a bit what my previous expertise on neurodegenerative diseases with the current model system, and I think it's kind of a unique mix of skill, and I'm hoping to sort of study what I've also been interested previously in this system.
Thank you. Oh, we've had another question. I thought that was almost the end. So how advanced are studies using these organoids generated from adult patients-induced pluripotent stem cells?
So, I mean, we're very, very early days of that, I would say, because there's now several studies on cerebral organoids. So that's been done, even to look at microcephaly and other diseases, neurodevelopmental diseases. But for Alzheimer and neurodegenerative diseases, I think we are still on quite early days, although there's some groups now that are starting to generate cerebral organoids. For example, Selina Ray at UCL is generating cerebral organoids with IPS from familial Alzheimer's disease patients, and we are also collaborating in order to look at the choroplexus. So there's some groups there starting to do this work now.
So I think that's all of the questions that we've had, unless anyone has any burning questions they'd like to send through now to ask Laura. But if you'd like to take up kind of project-specific questions, perhaps you could contact Laura directly after the webinar.
Thank you.
Great, thanks Katie very much for hosting those questions, and Laura, thanks very much for answering all of those. I'm just going to wrap up the session today just by thanking Laura and asking her to pass on congratulations to all the team that are involved in doing this work. So fantastic piece of work and great presentation. We're really pleased that you've won the prize. So thank you very much.
Thank you very much.
Thanks also again to GSK for ongoing support and making this happen. And thanks to everyone who behind the scenes and on Zoom has made this run so smoothly. We're going to make the webinar available on the NC3R's website in the next few days and have a press release to announce the award. So just leaves me to thank you all very much for attending. I think we had 130 or so people online. So it's great to see that level of interest in the prize and the ongoing work in the 3Rs area. So thanks very much, and I hope you have a nice evening.